Hi guys, it's Mrs. Allgood. Today we are going to talk about seed germination. I usually show several cool videos of seeds germinating, like, I don't know if it's real time or if it's sped up a little bit, and normally I can kind of talk you through there and what you're seeing. I can't do that this time. I'm afraid that if I embed it in our notes, that YouTube will block it, and then I'll have to record the lecture all over again. So please watch the videos that I post. It'll just give you a better idea about what we're talking about today. So this is not ideal. I wish I was in the classroom teaching this to you, but because we can't be there, I need you to make sure that you not only fill in the notes to this lecture, but that you actually watch the videos that I'll post that go along with it. They're not very long. They're not boring. It's just showing you the process of a seed germinating. Okay, so before we get into how seeds can germinate, we've got to look at some of the parts of a seed. Okay, so you might want to fill in your blanks here. This place that I kind of showed with an orange circle surrounded by black, that is the place where two cotyledons are joined together. Remember that we're looking at a dicot seed here. Remember that cotyledons are seed leaves, and in something like a bean, the cotyledon fills up most of the two halves of a bean. So, what you've got above where the two cotyledons are joined together is the epicotyl, because the prefix epi means above, okay? So epi is above, the epicotyl is above where the two cotyledons are joined together. Remember that the prefix hypo means below, so the hypocotyl is below where the two cotyledons are joined together. And cotyledons, remember, are seed leaves. And in dicots, they're basically going to absorb all the nutrients from the endosperm that we get as a result of the double fertilization that we talked about last week. Okay, the radicle, or if we were in the 80s, we would say radical, okay? It's spelled different. But radicle here, radicle is your embryonic root. So as you watch the videos that I post, most of them are from YouTube, as you watch those videos, keep in mind that the radicle, the embryonic root, is going to be the first thing that emerges from that germinating seed. Okay? Remember that in here, we are doing more than just memorizing. We want to truly understand this. So if you're just trying to fill in the notes, then just fill in the notes, but it's going to be hard for you to complete the quizzes and complete the test whenever we have one if you don't really understand. So I want you to watch those supplemental videos and just keep in mind that the radicle is your embryonic root and it's the first thing to emerge. And then these are just the young leaves. So usually when a seedling emerges from a seed, you're going to have two little leaves that will be the first ones that can do photosynthesis. And what's kind of cool about those is they usually look different than the regular leaves on the plant. They're not usually shaped the same way. They are just the leaves that came from the seedling. All right, let's talk a little bit more about seed structure and dicots. So this would be the place where the cotyledons are joined together. So the epicotyl would be above that as well as the young leaves. Then you have the hypocotyl and the radicle. Okay. So below where the two cotyledons are joined together, in other words, when you say cotyledons, just think of it as the two halves of a bean. Okay, below that you have two things. You've got the hypocotyl, which is going to become the stem that supports the plumule. The plumule is like the young leaves, okay? And then the radicle, okay? The radicle is the embryonic root. So hypocotyl is going to eventually become the stem. Radical is going to become the root. All right, above where the two cotyledons are joined together, you have the epicotyl, which becomes the shoot tip. And then you have your young leaves, your first little leaves that can do photosynthesis, but of course we'll have many more after that. And of course, if I ever go too fast, please just feel free to pause the video. All right, so what we were just talking about was seed structure in dicots. So let's look at seed structure in monocots. Monocot embryos only have one cotyledon. Remember, we talked about the corn kernel with that and that little white piece that's inside the corn kernel, and it does stay separate from the endosperm. Monocots still have a hypocotyl, a radical, and an epicotyl. It just looks different in them. We're going to focus mainly on dicot seed germination, and we'll focus a little bit on monocot seed germination. Now, a structure that monocot seeds have that dicot seeds don't is they have something called the coleoptile, okay? And that surrounds and protects the embryonic shoot. The way you're going to know the coleoptile is when a seed germinates and it starts to emerge through the soil, this is what's going to emerge through the soil from a monocot seed. It's going to look very much like a spear, okay, penetrating the soil. Whereas in dicots, what we see penetrating the soil boundary is going to be the hypocotyl hook, which is going to look kind of like an elbow. But in a monocot, it's going to look like a spear penetrating the soil when a monocot seed germinates. Okay, let's get into steps of seed germination. To me, this is very straightforward. So I would try to do more understanding than memorizing, but it's really not that complicated. But you've got to make sure you're looking at the picture. Okay. Back in the fall, when we did our cell respiration lab with our peas and beans, remember with the biochambers and the Vernier sensors, I had to germinate some seeds for you. So all I did, if you ever want to germinate a seed at home, you can't use something frozen or canned. That won't work. But you can use dried beans if you have those in a bag at home. Those seem to work the best, like kidney beans, lima beans, something like that. Black beans even. It just can't come from a can. It can't be frozen. But the first thing that you do is you take those peas or beans and you just put them in a container and flood it with water. Okay, just let them soak in water for about 24 hours. After 24 hours is over, you drain the excess water and then you wet some paper towels and you wrap those peas or beans in paper towels, and you can wait anywhere from 24 to 48 hours and you'll see those seeds germinate. So you don't have to even go buy seeds from a store. If you have any dried beans at home, like black beans, pinto beans, something like that, it works great for this. So the reason I tell you that is so you realize that the first step in order for a seed to germinate is imbibition. Okay, when you imbibe, imbibe means you're drinking something. So imbibition is the uptake of water by the seed. Okay, so that will cause the seed to expand and the seed coat to rupture. Okay, so when the seed coat ruptures, that's going to cause the embryo to release gibberellins, which is one of our plant hormones that we'll talk about tomorrow, and they diffuse into the alurone layer, which is a layer that's just inside the seed coat. Now, if I were to just tell you that, I think it would probably make no sense at all. But that's why I give you these pictures, okay? So this is a little seed, okay, and it has already imbibed. It's already taken in water. So what the embryo is doing, this is our embryo here, it is releasing gibberellins, which are these little greenish, grayish dots, I guess greenish dots that are being produced by the embryo. They're going past this orange layer, whatever that is, and they are going to this whitish color layer right inside the seed coat, which is called the alurone layer. So as the embryo grows, it releases gibberellins, which diffuse across to the alurone layer, which is just inside the seed coat. Okay, this is going to trigger the alurone layer to secrete something of its own. It's going to secrete amylase enzymes, which move into the endosperm. Okay, so if we go back, embryo releases gibberellins, which go to the alurone layer, this white layer. Well, when that happens, that's going to cause the alurone layer to start to secrete amylase enzymes, which are these little pink magenta color ones or purple color dots here. Remember, they're being produced by this white layer, which is the alurone layer. And notice that they're going to this orange section. This orange section is endosperm. An endosperm is made by double fertilization, and it is full of starch. Okay, so embryo releases gibberellins. That goes to the alurone layer. Alurone layer secretes amylase enzymes. Amylase enzymes break down the starches in that endosperm. Okay, those carbohydrates, remember that's the orange part, that's going to serve as nutrients for the growing embryo so that the embryo can grow even more. And so what you're going to see is you're going to see the embryonic root or the radicle come out first. And then you will see the hypocotyl, which is the stem, also emerge. So the first organ to emerge from the germinating seed is the radicle. And so in the videos that I'll show you, they're not really animations. I want you to look for that. I want you to see that the radicle is what's coming out first, and you'll probably see some root hairs associated with that. Okay, let's take this little by little. So next, the shoot tip must break through the soil surface. Now, that's going to look different in monocots and dicots. So if maybe your family has decided that they want to start a garden since they might be at home and might have time to and might have the land to, I want you to pay attention to those little seeds as structures start to break through the soil. Okay, so what we're going to talk about first is what's going to emerge through the soil if it's a dicot seed. Okay. So in dicots, the hypocotyl, which remember is kind of like the stem, ends up forming a hook-like structure. So we either call it the hypocotyl hook or the apical hook. Okay, and there's your little hook right there. Okay, and so to me, whenever a dicot seedling emerges through the soil, it's going to look like somebody sticking up an elbow and poking through the soil surface. All right, in monocots, that's not what you get. You don't get a hook or elbow looking structure. You get something that kind of looks like a spear, and that is the coleoptile. Okay, and it comes through the surface kind of like if you were to put your hands together and put them above your head like you were going to dive into a swimming pool. That's what the coleoptile looks like. So even if you didn't know these crops that you were planting and whether or not they were a monocot or dicot, you should be able to tell what they are by what emerges through the soil. Okay, hold on one second. Okay, guys, just a little bit more left, and then I'll get this uploaded, and I'll get those other videos posted. So in response to light, the hypocotyl straightens. So remember, it comes through the soil as a hook, but then light allows it to straighten out. The cotyledons separate, and the delicate epicotyl is going to spread its first true leaves. So here's what you have to think about. When a seedling is still in the soil, okay, it's getting the nutrients it needs to grow from the endosperm. Because if it's still in the soil, it can't do photosynthesis yet. But once it gets above the soil and can spread out those leaves, then it can photosynthesize. All right, so this is what you're going to see in some of the videos I'll show you. There's your hypocotyl hook. Okay, the two halves, if this is like a bean plant, these are your cotyledons. Okay, the cotyledons are going to shrivel and fall off because all their food reserves have been exhausted. That little embryo has used up all the endosperm that's in the cotyledons, and so you're going to see the cotyledons fall off. Remember, this is the hypocotyl. Okay, it's the stem that leads you up to where the cotyledons are, but above the cotyledons, remember that's what epi means, above the cotyledons will be your epicotyl, and that will connect to your first leaves, and then also your apical bud at the top. So as you watch some of these animations, I encourage you to look for, you're going to see one where the cotyledons fall off. You're going to see that hypocotyl hook, and I'll also show you some monocot ones as well. All right, in monocot, so I know I at least have one monocot one with corn. The coleoptile, again, is kind of like a spear coming through the soil. And then if you think of something like corn, you know, this is a corn kernel, that's our seed. And then you're going to have your leaves come that can start to photosynthesize. Okay, so I think that is it for the notes for today. Notes are fairly short. Like I said, notes are going to end up being about 17 and a half, 18 minutes long. So the time that you would have spent writing down stuff and listening to me, I need you to spend watching those other animations and trying to pick out those structures that we talked about today. In a dicot, you're going to, well, in either of them, you can pick out the radical and the hypocotyl. In the dicot, you're going to look for that hypocotyl hook, and in the monocot, you're going to look for the coleoptile. All right, see you later, guys.